Good day, this is Professor Will, CBMD PhD. Today is Tuesday, December 8, 2020. It is 12:22 and 22 seconds p.m. Eastern Standard Time. And this is a continuation of my microbiological research, particularly fake peptides. So let's hop to it. Useful fake peptides, Oliguria fuldomers, mimic peptides alpha helixes and effectively bind to drug targets. Date December 7, 2020. Source Wiley. Summary. Some useful drugs consist of peptides acting on their protein targets. To make them more efficient and stable, scientists have found a way to replace crucial segments of the peptides with uredo units. These oligurias, which are composed of urea, base units fold into a structure similar to that of peptides. Oliguria-based fake peptides enhance the options for rational drug design, concludes a study. Several drugs are peptides that inhibit or activate the actions of certain proteins. To enhance their efficiency, scientists are investigating peptide mimics. Peptide mimics contain strands of small organic units that resemble amino acids, the building blocks of peptides, but are not identical to them. The rationale is that proteolytic enzymes will less likely attack such fake peptide strands, so the drugs would be more effective. However, the synthetic strands called the oligomers must fold into the structure of the original peptide to bind to its target protein properly. These oligurias fold into a helix, one of the hallmark structures of peptides. However, there are slight differences. Oliguria helixes have fewer residues per turn, a smaller rise per turn, and a larger diameter than the original peptide alpha helix. To determine whether oligurias could mimic real-time peptide structures, the researchers prepared peptide oliguria hybrids and investigated their binding to target proteins. One of the targets, MDM2, is a natural regulator of the tumor suppressor protein p53. The other one, VDR, is a protein required in the regulation of cell growth, immunity, and other biological functions. For the MDM2 binding peptide mimic, the researchers prepared hybrids by replacing 3 terminal key amino acids with uredo units. For the VDR binding peptide mimic, they replaced the middle amino acid segment with the uredo units. After some rounds of optimization, the authors found hybrid structures with high binding affinities. The binding affinities were similar to those of the original peptides. Moreover, X-ray analysis revealed that the hybrid structures adopted a regular helical structure. However, the helixes were still a bit wider and had larger spaces between the side chains along the oliguria backbone than those of natural peptides. Peptide oliguria hybrids are expected to resist proteolytic degradation, an important goal in medicinal chemistry. Another advantage is that they allow more modifications. Alpha amino acids can be substituted at 2 positions, but uredo units have 1 site more. This means that hybrid peptide oliguria drugs offer more options for optimization. Good day, this is Professor Will, CBMD PhD, on microbiology, computer science, and bioinformatics. Well, hope you enjoyed listening. Until next time, have a great day.